Okay, so I'm going to share first web browser. As always, if you have a question, please put it in the chat function. So this is what Canvas looks like for you guys. Again, a few due dates that are coming up that I want you to be aware of. If you go to the assignments page, so the ones that you have coming up, again, your discussion questions for the week, you need one of them has to answer these questions, nervous system questions part 2. The other one has to be the things you found interesting or confusing week 2. So those are your 2 discussion questions or discussion posts for this week. You have a worksheet about the different types of glia that's going to be due April 10th, which is Friday at midnight. And then next week we have homework 5, which is due at our class time at 9:05. Second round of discussion questions, as well as there'll be a new things you find interesting post. And then there is a case study that I want you guys to do. We would have done it in class next week, but we don't meet as class time. So instead, we'll do it for out of class. And then the cranial nerve worksheet. And then we will start chapter 8, which is our senses. So we're getting to the mid-end part here of chapter 7. Okay, so with that, do people have any questions about what is due this week or next week? Another reminder that Friday is Good Friday, so we won't be having a WebEx class. You have that GLIA worksheet due and your discussion boards, but otherwise there won't be any work for Friday. Any questions people have about what is coming up? Okay, I'm going to switch gears and we're going to jump into the PowerPoint here. Okay, so what we talked about last time are reflexes and then the different parts of the brain. And so what different parts of the brain do. As a reminder, you need to know where you can find, like on a picture, the 4 lobes of the brain. It should be pretty straightforward because, again, those 4 lobes match with the bone. So we have the frontal lobe, then we have the parietal lobe on top, the occipital lobe in the back, and the temporal lobe over your temples. Not only do you need to know where those 4 lobes are, you need to know the functions, what functions are assigned to each of those lobes. So if you see like a list of functions, you need to be able to tell me what lobe of the brain they would go in. For the deeper brain structures that we talked about, the midbrain, the diencephalon, the brainstem, those sorts of structures, you just need to know what they do. So you don't need to know where they are on a visual map. You need to know which grouping they go into, but you don't need to know where they are on a visual map of the brain. You just need to know the function. So we're going to jump into part 4 here and answer a question that we've been thinking about this for a while. So we've been thinking about why we need to protect neurons. What makes neurons so special? Remember, neurons are the signaling cells in the brain. They are those electrical cells, and they go throughout the body in our central and our peripheral nervous system. And neurons are special because they are post-mitotic. Neurons are delicate. They cannot reproduce anymore, so they can't replicate. They can't fix themselves by making more neurons. So if you have a brain injury or a spinal cord injury, you're not going to be able to repair that by filling the gap via mitosis in the same way that you would with skin. Neurons are also special because they have a very high energy demand. So as we looked at the action potential, there's a lot going on there. There are a lot of enzymes or receptors working, and all of that takes energy. So the steps of the action potential, the voltage-gated sodium channels and the voltage-gated potassium channels, they use the energy that's stored in those gradients, right? The high amount of sodium that needs to be present outside the cell and the high amount of potassium that's present inside the cell, those gradients. But to set up those gradients, that takes the sodium-potassium pump, which uses ATP. So neurons use a lot of ATP. In fact, when you're taking an exam or you're studying hard, an hour of intensive focus will burn the same amount of calories as running a mile. And that's because your brain is using all of that glucose to make ATP to keep these gradients up. And so neurons are post-mitotic. They have a high energy demand. And so they're also very important functionally. As we know, all of our decisions, all of our actions, they're controlled by the nervous system. This is where we integrate all of our stimuli. So they have an important job, they use a lot of energy, they can't repair themselves. And all of that leads to the body spending a lot of energy and a lot of structures to help protect these delicate cells. So we protect them in a few different ways. We use bone to protect our central nervous system in the form of our skull around our brain and the vertebrae around our spinal cord. We also cover them in layers of membranes called meninges, which I'll talk about. And finally, they're bathed in a fluid. This is called the cerebral spinal fluid, and the fluid also provides some cushioning. The other way that we've talked about already are the glia cells. The glia cells form that blood-brain barrier with the astrocytes. They have cushioning and supporting roles themselves, and so glia are another way that we protect our nervous system that we've already discussed. But let's dive into a few of these other ways of protecting our nervous system, and the focus is really on the central nervous system because it is the integration center. It has the most important function compared to the peripheral nervous system. So we're going to focus on how we protect this crucial part of our nervous system, our brain and our spinal cord. So we cover them all in meninges. The bones we've talked about when we talked about the bone chapter, but meninges are layers of membranes that cover the central nervous system. So if you start with your scalp, you have the skin of your scalp, then you're going to have a periosteum layer, which separates the skin from the bone. Then you'll go into that protective bone. In this case, we're talking about the brain. So we have our skull, we have all of those cranial and facial bones that make up the skull. And then you have 3 layers of meninges. You've got your dura mater, your arachnoid matter, and your pia mater. So you need to know those 3 layers of meninges. They make the acronym DAC if you're going from outside in, dura, arachnoid, and pia layer. And then under that is where you're going to have our dural subarachnoid space right in between the layers. And under all of that is where you're going to have your CSF, so your cerebral spinal fluid, which is going to be bathing the brain. So the CSF, or the cerebral spinal fluid, acts as like a cushion. It's continually made in the region of the choroid plexuses, and it's made from blood. And so they're going to take bits of the blood and they're going to make it into this fluid that's going to go through the roof of the ventricles. Remember, the ventricles are these holes that you have in the middle of your brain. So the cerebral spinal fluid is going to go from the roof of the ventricles and circulate through the ventricles and down your spinal cord. And so if you were to look at the brain, it would appear like it was just gently floating in fluid. And this, again, is a cushioning. So if you stop short and you hit your head on something, for example, your brain is going to move forward, but it's going to swish back in part because of all that fluid holding it around. So it adds that extra protective layer. And this is the lateral view, the side view of the ventricles. We've got lateral ventricles, your 3rd ventricle and your 4th ventricle, and then the cerebral aqueduct, which is going to connect down into the spinal cord. And the fluid is going to move in a stereotypical pattern. Remember, the ventricles are lined with those glia cells, the epidermal cells, that have the cilia. So the cilia is going to wave, and we're going to move the fluid through our ventricles. And so if we follow that path, we start with the choroid plexuses making the CSF here shown in number 1, and that CSF is going to flow through our ventricles into the subarachnoid space in between that arachnoid meninge layer. Then some of it is going to enter the central canal of our spinal cord and go down. The rest of it will flow through the subarachnoid space and then be absorbed via the dural venous sinuses into the dural venous sinuses via the villi of that arachnoid layer. So there are little protrusions into the meninges where we can absorb some CSF and it will go back into circulation into the rest of the body, but the rest of it will continue to bathe the brain. And CSF, the other thing you want to know about CSF is it will go down your spinal cord. And so if you've ever heard of someone having to get a spinal tap before or a lumbar puncture, lumbar being a lumbar region of your spine, that's generally where they'll stick a needle in between the vertebrae and they'll remove some of that cerebral spinal fluid. And so some reasons why they do that, one is maybe they want to test and see what is in the cerebral spinal fluid. Again, They want to see what's circulating around in our central nervous system. They might have to do a lumbar puncture or a spinal tap to get some of that fluid. They also might do it to relieve pressure. So this fluid is circulating around the brain, but if the brain starts to swell, all of that fluid has nowhere to go, right? And so that might create an increase in intracranial pressure or brain pressure. And so what they can do is they can drain some of that fluid out from the spinal cord, and that will relieve some of the pressure that's on the brain. Okay, so the blood-brain barrier, we talked about this before, but the blood-brain barrier is our last protective measure. It is the ultimate gatekeeper. So again, this is made from the barrier between our blood vessels and the astrocytes, those glia that help form those barrier between our brain and our body. And again, we have blood vessels, we have capillary cells, and they're connected with tight junctions. So that means that things are not going to flow through. Okay, so water, glucose, and amino acids are the only diffusible water-soluble substances. So the majority of things like ions, for example, or large molecules, toxins, that's going to stay in the capillaries. And we have special transporters that will instead just bring in what the brain needs. So the brain needs glucose, the brain needs amino acids, and the brain needs some water. And the transport of glucose is especially important because your brain can only use glucose, whereas other parts of your body can undergo different processes to turn proteins or fats into energy. Your brain can't do that. Your brain only has glucose transporters, and so it can only take in straight sugar. Which is why if you get hypoglycemic or you have low blood sugar, you're going to start to feel kind of lightheaded and woozy. That's because your brain isn't getting the glucose that it needs to do its functions. So if you feel that way, you should eat a snack. You should eat like a Snickers bar if you eat Snickers bars or whatever that commercial tells you to do. Fat-soluble things, as with all membranes, fat-soluble molecules are going to easily diffuse in. And then gases also don't need a transporter. They are easily going to go through things like oxygen, carbon dioxide. Laughing gas, very easy to get into the brain, very quick to get into the brain, again, because it's a gas. And so if you work in the pharmaceutical industry and you're interested in designing drugs for the brain, you have to keep in mind that the vast majority of things don't get into the brain. And so it either needs to be fat soluble, or it needs to be a gas, or it needs to be similar enough to an amino acid or glucose that it can be transported in, because otherwise it's going to stay in the blood and it's not going to get into the brain. So this is a huge roadblock in the pharmaceutical industry when we're trying to get drugs that target the central nervous system. Okay, so question you might see is describe ways that you would protect our central nervous system. You need to talk about things like bones, meninges, fluid, blood-brain barrier, glia cells forming that blood-brain barrier. Okay, all of these key ways that we protect our central nervous system. Are there any questions so far? You can type them in the chat while I get a little water. Okay, so we're going to keep moving. We're going to talk about some diseases. So this is a picture of the Patriots playing some excellent football here. And the reason I'm showing this is because one of the more common brain dysfunctions, or that's gotten a lot of media attention, are traumatic brain injuries, or TBIs. And again, all of these issues that arise from brain disorders are because neurons are postmitotic. So the reason we care about brain injury so much is because the brain cannot repair itself in the same way that a lot of other organs can. And so traumatic brain injury, repeated brain injuries can have really long-term lasting issues. So we use words like concussion, which is going to be a brief loss of consciousness. There are all sorts of concussion protocols and standards to check to see what's a real concussion and to grade a concussion. But generally, it involves a brief loss of consciousness and some confusion and some issues that arise right after the event and can persist depending on how bad the concussion are for weeks, months at a time after. A contusion is just a bruise. So a contusion, when we're talking about brain injuries, a lot of times it's the force of your brain hitting your skull. So if you dive and you hit the ground really hard, or if you're driving and you hit the brakes really hard, your brain is going to keep moving forward even as your skull has stopped. And so your brain will hit your skull. And if it's bad, it can cause a contusion or a bruise. And if it's really bad, you can get a concussion. Another term that's thrown around, another symptom of TBI, are intracranial hemorrhage. So this is a brain bleed. So bad brain injuries can cause brain bleeds, where the little blood vessels in your brain start to leak blood. This is a problem, again, because we keep the blood in our brain under very tight wraps, right? It's sealed away with that blood-brain barrier, and the fluid in your brain is not blood, it's CSF. And so brain bleeds are bad, not just because there's blood, but that blood is pushing away the CSF, and it's harder to clean up than in other areas of the body. So brain bleeds are more serious in that regard. And finally, we'll talk about cerebral edema, which is a swelling of the brain. As I said before, the brain is in a closed system with the CSF, and so if the brain starts to swell, it can create this increased pressure because the fluid, your CSF, has nowhere to go. And so that needs to either be relieved, usually the spinal tap, otherwise known as a lumbar puncture. We talked a little bit with aphasia about stroke. Strokes are the third leading cause of death in the United States. The other name for stroke is cerebrovascular accident, or CVA. So in the medical field, you'll see strokes abbreviated as CVA. And some of the main symptoms are paralysis. You can get a hemiplegia, which is half of the body could go and have no motor function, which is the paralysis. You can't move that half the body. And again, it depends on your dominant side. So if you are right-handed, the left side of your brain is going to control a lot of the left side will control the motor function on the right side of your body, and the right side of your brain will control the motor function on the left side of your body. So depending on where the stroke is, that will determine the motor deficit that you have. You also have language deficits. Again, we call those aphasias. Those will vary depending on where it is. And this is all caused by what we call a transient ischemic attack. So usually it's a blood clot that travels up into your brain and cuts off the oxygen supply to that region. And if it's small, the body will just dislodge it itself, and those small strokes are not as big a deal. If it's bad, that can cause a rupture in that area, and it will kill off all of the brain tissue right around where that happens. If it's really big, it will have a much larger area of damage. So strokes, the severity depends on how bad the blood clot is. Is it small? Is it big? Are you able to get to a hospital in time? If you suspect someone is having a stroke, you need to get medical attention right away, because there are drugs that they can be given that will help dislodge that blood clot before it bursts. So they need to very quickly be brought to medical attention. Signs of a stroke are, again, can be like a drooping face on one side, usually confusion, and then language deficit, so a slurred speech or nonsensical speech are some of the major signs that a person is having a stroke. Okay. So we're going to move in our central nervous system from focusing on the brain to talking about the spinal cord. So your spinal cord is the information highway of your body. It's what connects your peripheral nervous system to your brain. It's about 17 inches long. It runs from your skull. It doesn't go all the way down. It stops at around L1, L2 in your lumbar region. It is also surrounded by a layer of meninges, or 3 layers of meninges. And coming out of your spine, you have 31 pairs of spinal nerves. At the very end, the spinal cord becomes a region that's just nerves that we call the cauda equina region. This is named because I've never ridden a horse before, but people tell me if you ride a horse, the inside of your legs can get sore. Those muscles on the inside of your legs, your adductor muscles especially, can become sore. Well, the very end of your spine in the cauda equina region is what innervates or gives sensation and motor function to the inner part of your legs. And so if you get damage to the end of your spine, if you get damage to the cauda equina region, you'll go numb and be unable to move the inner part of your legs. And so that's where they came up with the name equina. So think horseback riding, what becomes sore, inner part of the legs. Okay, so this is a view of the spine. You have spinal nerves, and they're named for the spinal region that they come out of. So the ones that are in the cervical region by your neck are called cervical spinal nerves. Thoracic in the chest are the thoracic nerves. And then they end with the lumbar nerves, and the very tail becomes cauda equina region, which will shoot off a few sacral spinal nerves. The spinal nerves are, the spinal cord is thicker in two regions, and it's thicker because a lot of nerves come out of those two regions. So it has a cervical enlargement and a lumbar enlargement. The cervical enlargement is because it's going to send out all of the nerves for your arms, and the lumbar enlargement sends out all of the nerves for your legs, or the majority of nerves for your legs. And so those regions are bigger in your spinal cord because they have to fit all of those nerves branching out to those larger areas of your body. If we look at your spinal cord, we can break it if we do a slice. We look at it and we see that there are two different regions. There is gray matter, it looks gray, versus white matter that looks white. We have a dorsal horn and a ventral horn, dorsal being back, ventral being front. In the dorsal horn, we're going to have the cell bodies of the entering sensory neurons. So the sensory information is going to be on the dorsal side, and the ventral side is going to be the motor. And they will send out roots, which are going to fuse and become spinal nerves. So in the spinal cord, we separate sensory and motor function. This is why some spinal cord injuries, if it's not a full bisecting injury, so if it doesn't completely break the spinal cord, the patient might have either sensory deficits or motor deficits. So if they just damage the dorsal part of their spine, then they're going to just have issues with sensory from that level and below. So if they get lumbar damage, for example, in the dorsal part of their spinal cord, then they wouldn't be able to feel their legs. They would still, however, be able to move their legs. And this is really interesting because, again, that motor part is in the ventral part of the spine. So the sensory, that's damaged. They can no longer feel their legs. But if the motor is intact, they can still move them. And the reverse is true. So if they damage the ventral part, they could still feel their legs, but they wouldn't be able to move them. And so with spinal injuries, you need to pay attention to what part is damaged or whether both are completely damaged. This is again what it looks like. You see the gray matter and the white matter. The sensory and the motor neurons are going to run off, and then sometimes they come together and they form that spinal nerve. So the spinal nerve is going to have both sensory and motor input, sensory going up the spinal cord, motor input going out from the spinal cord. They'll come together in the spinal nerve, but they'll branch when they enter into the spinal cord itself. So again, the white matter in the spinal cord is made of those fiber tracts. We have got different columns. The sensory tract is an afferent tract. It approaches the spinal cord and brain. And then the motor tract is efferent or exiting. It's going to leave from the brain down the spinal cord. And this is, again, what that looks like. We're going to get integration through the thalamus is that relay station. It's going to go up to different parts of the cortex and decide what to do and send a response. So we start from the sensory receptor. We have an afferent or approaching sensory neuron that's going to go into the spinal cord, go up into the brain. Decision will be made. Movement will come down the efferent pathway and go out a motor neuron. Questions to consider are intraneural questions. So we're gonna, with that, we'll talk a little bit about the peripheral nervous system. For this part, we'll talk about the general structure of our peripheral nerves. We'll identify cranial nerves and list their function, and then we'll talk about our sympathetic and parasympathetic system. So what I want you to know for here, again, because we started talking about the spinal nerves, is that nerves are bundles of fibers, and most of them are mixed. So mixed nerves are all of our spinal nerves, so everything coming out of your spine has sensory fibers and motor fibers. Sensory coming from the body up to the spinal cord, motor fibers coming down from the spinal cord. But you have some nerves in your body that are also just sensory or just motor. So all of your spinal nerves have both, but some of the other nerves in your body will just have one or the other. Okay, that is where we're going to leave it for today. Any questions people have? Any questions about what you need to be working on? The material we just covered. We finished the central nervous system. We're just about to start the peripheral nervous system. Okay, I'm going to stop the recording here.